Dear students, welcome to my YouTube channel. So in this video we will study about the Plasmodium vivax in detail. So I am following the two books. First we have Zoology Applied, Zoology VSC third year का है, and second we will follow the medical parasitology. Some topics which are not present in this we will add from medical parasitology, like some diagrams and some topics which are not included in this. Okay, so mainly we will focus on this book, that is the modern VSC third year. So in this video we will study about distribution, habitat, habits, and main is the life cycle of Plasmodium vivax, which is the diagenetic in nature. That means it has the two hosts, a sexual cycle in man, okay, and second sexual cycle of Plasmodium in mosquito. So we will discuss in detail mainly life cycle. Next we will discuss about the pathogenicity, symptoms, prevention, and last therapy. Okay, so let's discuss with the distribution. Distribution, it is the cosmopolitan types of parasite. That means it has worldwide distribution. It widespread in tropical and temperate countries. If we talk about Plasmodium vivax, so it is the less virulent. Virulent means infection, so it causes less infection as comparison to Plasmodium falciparum, because this Plasmodium falciparum species cause the death. Okay, so even Plasmodium vivax also causes death, but rare cases. Okay, so there are many countries like Asia, South America, which has the highest distributions of the Plasmodium vivax, and other countries like India, Sri Lanka, Bangladesh, Nepal, Pakistan are worst affected. If we talk about in India, so in India they are especially present in Bihar and Uttar Pradesh. Okay, next we have habitat. So habitat is the endoparasite in nature. Endoparasite means that live inside the body. In previous video we studied about the life cycle of Ancylostoma terminale. So they have also the endoparasite in nature. That means it live inside the body, and mainly they are the gut endoparasite. That means they live in the intestine of man, cat, and dog. But in this case, Plasmodium vivax found in the blood of man. Okay, so intracellular in RBCs and the parenchymal cells or liver. So location is the intracellular in RBCs. We will discuss in the structures in detail. Okay, so intracellular in RBCs and parenchymal cells of liver. So it is also found in the alimentary canals, salivary glands of female Anopheles mosquito. Next we have habits. So being intracellular parasite, it depends for all the vital activities on host. So this Plasmodium vivax depends on the host, that is the man and mosquito, for all the important activities. Next we have the life cycle, which is the most important and in more detail. Okay, so in this we all know that Plasmodium vivax is the diagenetic in nature. That means it have the definite host and the intermediate host. Definite host and intermediate host. So this structure we will discuss in this book, that is the medical parasitology. Okay, so you can also follow this book. So this is the life cycle of Plasmodium vivax. Okay, so this is easy structure, easy understand as comparison to this. Okay, so this structure is something in detail. Okay, so you can draw anyone. So if we talk about the definite host, and we all know that Plasmodium vivax have two hosts. Okay, so it has the definitive host, male mosquito, and intermediate host is the humans, or we can say monkeys. Okay, so life cycle of Plasmodium occurs in three phases, that is the Sachirogony, Gamogony, Sporogony. In human body, these parasites multiply asexual. So we all know that asexual cycle, and this occurs in the man. It is further divided into the exoerythrocytic cycle or erythrocytic And in exoerythrocytic, it occurs in the human ovulation infections. We will discuss the structure of sporozoites and liver succiogony, which is further divided into the pre-exo and pre-patent incubation period. Okay, so let's discuss with the exocycle and succiogony in man. So this is the man. Okay, so this human. Okay, so this is the life cycle in sexual cycle in mosquito and asexual in the human. So let's discuss with the first, that is the exoerythrocytic. Next we will discuss about the erythrocytic. Okay, it has the two phase. First we have exoerythrocytic succiogony. So it is further divided into the three stages, that is the human inoculation. Next we will discuss about the sporozoid structure and liver succiogony. Okay, so this liver succiogony will further divide it into three stages, and last in this we will discuss about the erythrocytic. Okay, this is the second. So let's discuss with the first one, that is the exoerythrocytic succiogony. First we have human inoculation or infection. A healthy person acquires infection when an infected female, that is the Anopheles mosquito, containing the infective stage of parasite in their salivary glands. So, supposed if there is a mosquito that not contains the infective stage of parasite, that is the sporozoid, they infect the man. So man will not infect. Why? Because the mosquito do not contain the infective stage of parasite. But if the mosquito contains this infective stage of parasite, that is the sporozoid, and they infect the human, okay, so then man will infected by the Plasmodium vivax. Okay, so they feed upon the human blood which they suck, usually during the night, by means of their pursing and sucking mouth parts. So this is the mouth parts nature of the mandibles. So the mosquito puncture the host skin. This mosquito punctures the host skin. Okay, so by its proboscis it present in their mouth. So by the proboscis it punctures the host skin and first introduce some saliva into the wound. If the saliva contains the infective parasite, that it will infect. So this parasite, suppose this mosquito contains the infective stage of parasite and punctures the host skin, that is the human, and it inoculates the infection, or we can say the saliva into the wound. So saliva contains the anticoagulant. Anticoagulants means which secretes the clotting of blood during the sucking. So the by the anticoagulants, that means blood will not clotting, because our blood property is clotting. So after the anticoagulant secrete, the blood will not clot. Okay, so parasite, the mosquito can easily suck the blood. Okay, so the saliva contains thousands of sporozoid, that is infective stage. So this infective stage injected into the humans. Okay, so this is the human inoculation infection. Easy. Okay, next we have structure of sporozoid. So this is the structure of sporozoid, ultra structure of sporozoid under the electron microscope. This is important. So we all know that this sporozoid is a spindle shape and slightly curved or sickle shaped body tapering at the end. It is a nucleate organism, that is it has the one nucleus and measuring the 11 to 12 micro in length and 0.5 to 1 micro in width. So each has covering of firm but elastic pellicle, okay, containing a longitudinal contractile microtubules for its wriggling movements. Its anterior end is marked by the minute aperture called the micropyle. Okay, this is the micropyle. So it has the apical cap of 3 or more connective rings and a pair of long slender secretory organelles open into the cap. So this is the main structure, we will draw it, and the main point, that is the mitochondrion pellicle, convoluted tubules, nucleus, micropyle, paired organelles, microtubules, so pellicle, apical, cap. So this will see main parts and will draw. Okay. Next we have the liver sachizogony. So within half an hour or after the inoculation into the human blood, all the sporozoites will disappear from the blood. Okay. Why? So as they reach into the liver, now from the blood they reach into the liver. Okay. So with the blood circulation, they make their out from the blood capillaries and enter into the liver cell. So after the blood, they come into the liver. So liver sachizogony have the two phase, that is the pre-erythrocytic phase and erythrocytic phase. Okay, but in this we will discuss the another phase, that is the pre-patent incubation periods. So in the pre-erythrocytic phase, you can see the structure as well. This is pre-erythrocytic cycle. Okay, so in the liver cell, the parasite becomes spherical and known as the cryptocytozoids. Okay, cryptocytozoids in the livers. So in the liver cell, the cryptozoids feeds on the cytoplasm and grows into the rounded structure, that is the pre-erythrocytic cytozoans, or also known as cryptocytozoans. So the later it divides into the thousand minutes crypto, that is the cryptomerozoites, by a special fission, that is the sachizogony. So in this, the in liver cell, the parasite of the plasmodium will become spherical. Okay, and you can see in this. Okay, it becomes the spherical and it will feed on the cytoplasm. Okay, so how they occur? They occur by the fission called the sachizogony. Okay, so the sachizogony called the exoerythrocytic because it occurs away from the blood. Exo means external, it does not occurs in blood and RBCs. Okay, so the sachizones and liver cells break up and they liberate the cryptomerozoites into the liver cyanoids. So these cryptomerozoites are immune to medicine and resistant of the host. So medicine does not affect due to the cryptomerozoites are immune to medicine and resistant. It has the resistance capabilities. Okay. So these cryptomerozoites are easily resistant to the medicines from these cyanoids. Some of these cryptomerozoites invade the fresh liver cell to continue sachizogony. So these the malarial parasite can remain dormant in the liver for days to years. So these malarial parasite remain in the liver. Why? Because it has the resistance capability to the medicine. So medicine does not affect. So they can easily survive in the liver for about days or we can say years. So because there are no symptoms of the plasmodium. Okay, and remaining undetectable in the test. And if we do the test of plasmodium vivax and there is a presence of this infective stage, still they will not show the plasmodium vivax. By because it has capability to resistance the medicines and it can dormant in livers for days to years. Okay, so these are called the hypnozoites, that is the sleeping organism. These allow relapse of malaria into 5 years after the elimination of red blood stays and the clinical cure. So it can stay approximate 5 years in the host. Okay, next we have the exoerythrocytic phase. Exoerythrocytic phase. Okay, so we are doing here liver sachizogony. So these three phase, that is the pre-erythrocytic phase, exoerythrocytic phase and pre-patent incubation period, these all are occurring in the blood. Okay, next will the erythrocytic, this is also occur in the liver. So in exoerythrocytic phase, cryptomerozoid, that is, okay, So these cryptomerozoites enter the fresh liver cells to become the metacryptozoites. This is the erythrocytic. Okay, merozoites enter into the bloodstream. So enter into the liver cell to become the metacryptozoites. Okay, and later there are two types, that is the micro and macro metacryptozoites. So these microcrypto uh, merozoites which pass into the bloodstream and invades the RBCs, it will enter in the RBCs and invade them. And the macromeracryptozoites which invades the fresh liver cells to continue the exoerythrocytic cycle. This micro will infect the blood RBCs, okay, and macro will infect the liver cells. Next we have pre-patent and incubation periods. So the pre-erythrocytic phase is completed in about 8 to 10 days. This interval between the inoculation and initiation of erythrocytic phase is called the pre-patent period. Next we have the erythrocytic schizogony. So this cycle of the liver schizogony, okay. So this cycle occurs in RBCs and begins when the micrometacryptozoites enter into the RBCs. We studied here micrometacryptozoites infects the RBCs, okay. So it includes the following stages, that is the trophozoite stage and signet ring stage. Next is the emboid stage, Sachizones, and next we will the last formation of gametocytes. Okay, so next we will discuss about this hexocycloplasmodium in mosquito. So in trophozoite stage, that is the inside the capsules, the parasite become rounded and grows during the growth period. It is called the trophozoites. So this is the structure of trophozoites. Next we have signet ring stage. So as the trophozoites grows in size, a large non-contractile vacuole appears in the centers. So this is the vacuoles appears in the centers and pushing the cytoplasm and the nucleus to a thin peripheral layer. Okay, so this stage is clinically referred as the signet ring stage. Next we have the emboid stage. Emboid stage, the signet ring trophozoites develop into the active emboid trophozoites. Now the trophozoites start feeding more actively upon the cytoplasm of the host RBCs with the help of pseudopodia, and it secretes out the digestive enzyme which liquefy the cytoplasm of RBCs by the partial digestion. So the hemoglobin is not digested, it feeds on the RBCs by the partial digestion, and it gets decomposed into a brownish black pigment, that is the hematine, okay, which lies in the cytoplasm of trophozoites. In about 36 hours, the young trophozoites grows into the adult and occupies almost the RBCs, okay. So these trophozoites grows into the adult in the emboid stage. Okay, so besides the plasmalemma, endoplasmic reticulum, mitochondria, um, food vacuoles and Golgi complex nucleus, etc., a concentric structure of unknown significance has been recently observed trophozoites by the electron microscopy by the Graham. So you just follow this, okay. This is the emboid stage. Next we have the suchizone, that is also called the mirons, okay. So the emboid trophozoites after active feedings grows into the large size, that is the suchizones we studied here, okay. So it now undergoes the suchizogony. Its nucleus undergoes repeated mitotic division. So in nucleus, mitotic division takes place in the suchizone space and forming the approximate 12 to 24 daughter nuclei which get arranged at the periphery and the cytoplasm masses surrounding them. Okay, so each cytoplasmic mass with one nucleus becomes an oval merozoites. They are shorter if we talk about the morphology. So they are the shorter and thicker than the sporozoites, and a part of cytoplasm of suchizone is not used in the formation of merozoites. Okay, so you just follow from this. They are shorter, thicker, not needs to write. Next we have the structuromerozoids. We will draw structuromerozoid, that's it. Next is the last, that is the formation of gametocytes. Okay, so after the number of erythrocytic cycle, some merozoites invade the RBCs and grows into the different types of rounded form called the gametocytes or the gametes. Okay, so it becomes the rounded shape called the gametocytes. So this shows the sexual dimorphism. That means it can easily identify, we can easily identify it's the male and female. Male, that is the microgametocytes, that will be smaller and clear cytoplasm and contain a large diffuse nucleus. Okay, as comparison to others, female will be larger in size, clear cytoplasm, it will be full laden cytoplasm, it is the large diffuse, and there will be small compact eccentric nucleus. So we can easily identify the male and female megametocytes, okay, and the microgametocytes. Microgametocytes for the male and megagametocytes for the female. Okay, so in this we will study the asexual cycle in man. Okay, so asexual cycle in man, we study the two phase, that is the exoerythrocytic stachydogony and the in short I just covered up asexual cycle in man. We did the exoerythrocytic and the erythrocytic stachydogony. So in exoerythrocytic, it is further divided into the another, that is the human inoculation. Okay, first is the human inoculation. Okay, we studied here, and next we will we studied the structure of sporozoites, and last is the liver stachydogony, that is the pre-erythrocytic phase, exoerythrocytic phase, pre-patent, and the incubation period we studied. In B part of the Earth's exocycle in man, we studied the erythrocytic stachydogony, in which we study the prophozoid stage, signet ring stage, emboid stage, stachyozones, and the formation of gametocytes. Okay, we studied it. Next is the very easy sexual cycle of Plasmodium in the mosquito. So sexual cycle in the mosquito, this is okay. We studied this, and now we will discuss this. Okay, sexual cycle of Plasmodium in the mosquito. So we will study the mainly two cycle or two phase, that is the sexual cycle and sporogony. And sexual cycle we will study here, very easy it is. Okay, it will we will study the gametogenesis, fertilization, ookinetes, and in second sporogony it is not divided into other parts, it will easily uh, definition. Okay, so next we will finished it the life cycle. Okay, so main is the structure. So structures we will draw in very simple and understanding. Okay, so what is the gametogenesis? We already studied in previous classes. Okay, gametogenesis means the development of two gametes, male and females. Okay, so we all know that male and females are dissimilar in nature. We can say it shows the sexual dimorphism, means it can easily identify, it's easily differentiate from the female or from the male. So such gametes are called the heterogametes. Hetero means different gametes. Okay, so next is the formation of male gametes, formation of female gametes. So the male gametes undergo a spermatogenesis. We all know that. So the nucleus of each rapidly divides by the A mitosis into the 6 to 8 haploid nuclei. So after the division, this is okay, microgametes microgametes, or we can say microgametocytes. Okay, so this undergo a spermatogenesis, and this their nucleus will rapidly divide while the A mitosis into the 6 or 8 nuclei, haploid, that will haploid. So the later it will migrate to the periphery, and the cytoplasm gather around the nuclei. So suddenly each cytoplasmic mass together with the nucleus shoots out from the surface of the microgametocytes in the form of 20 micro and the 25 micro long. So this is the sperm. So next we will the fertilization. So in the fertilization, this is the male gametes, or how the female gametes develop. So it develops, undergoes the maturation phase, and forms a single female gametes. or we can say the macrogametes or ovum. Okay, so in fertilization, the megagametes develop a small cytoplasmic projection, that is the cone of reception. Okay, so this cone of receptions on one side only. Okay, so the nucleus moves into the cone, the nucleus will be moved into it. Okay, the nucleus will move into the cone, and one male gametes penetrate the female gametes, okay, into this cone, like this. Okay, so the nuclear and cytoplasm of two gametes fuse to produce a zygote. Okay, so the zygotes are formed in the stomach of mosquito. We have studied here mosquito. So the zygotes are formed into the stomach of mosquito, and it forms about the 9 to 10 days after the blood meals. Next we have the ookinete. So ookinetes, the zygotes when formed is rounded and the motionless, we studied. Okay, after remaining inactive for about 24 hours, each zygote start elongated and undergoes certain changes that in about 9 to 10 hours it becomes the 15 micro and 22 micro long and 3 micro thick. So worm like, you can change the structure change. Okay, so worm like motile organism, that is the ookinete. So this is the motile, it can move easily, and these all are the inactive states. Okay, so next we have the sporogony. This is the sporogony. Okay, sporogony stage, so this will be last. So each oocyst undergo a sexual multiplication known as the sporogony. This is the asexual multiplication. Okay, so its nucleus, the nucleus divides repeatedly by the mitosis, forming a 10,000 minute protonuclei within the 2 to 3 days, and at the same time the cytoplasm develop the large vacuoles and takes a sponge like structure. So each cytoplasmic mass develop a large vacuoles, we can say the change. Okay, so it doubles into the large vacuoles and form the sponge like structure. So each cytoplasmic mass become elongated and spindle shaped. So these spindle shaped bodies are called the sporozoids. These are the sporozoids. Okay, these are the spindle shaped and called the sporozoids, and these sporozoids reach the salivary glands of the host and become lodged in its common salivary duct in the large members. So these salivary glands of a single infected mosquito may contain as many as approximate 2 lakh sporozoids in mosquito, and whole asexual, or we can say sexual cycle is completed within 10 to 20 days depending on the temperature. So thus the life cycle will be continue as it will inoculate it, and another in liver cytogony occur, and development of stages like trophozoite stage, signet ring stage, symbiote stage, cytogenes, and formation of gametocele. And further the sexual stages of the plasmodium and mosquito will take place in the mosquito, like sexual cycle and sporogony and formation of male and female gametes, and then fertilization takes place, ookinetes, and last will be sporogony, it produces the spindle shaped sporozoids. Okay, so this is the life cycle of the Plasmodium vivax. Okay, next we will discuss about the pathogenicity. So in this book, pathogenicity is included, but not other like symptom prevention and therapy is not included. Okay, so we will follow the this book. So this is the pathogenicity. Malaria is one of the most disease of mankind. So it is common in the tropical and subtropical countries. We studied here, like if we talk about India, so in India it is present in Bihar and Uttar Pradesh. So cause, next we have cause. We all know that the malaria is caused by toxic pigment, that is the hemozoin, which formed from the hemoglobins of the RBCs, which are when destroyed by developing the stage of malarial parasite. Okay, So we will follow this diagram only from this medical parasitology books. Next we will add from the applied biology. Okay, so we done. If you have any query or any topic from this book, you can comment. So its cause is the pigment hemozoin. It formed from the hemoglobins of RBCs, which are when destroyed by the developing stage of malaria parasite. So the Plasmodium is a cosmopolitan, that is the it shows the worldwide distribution as well as in tropical and subtropical countries, and it is a endoparasite in nature. We studied in later. Okay, so we don't need to explain. Next we have the types of malaria parasites and types of malaria. So there are different types of malaria parasite, that is the falciparum, Plasmodium vivax, whale, etc. Okay, so this is a table form, you can study it in it, its geographical distribution, patent period, and etc. Okay, but we don't need to go in detail. Main is the life cycle. Next we have the symptom of malaria, that is the cold stage. Chilling and shivering will take place, and next high fevers can occur, and faster respiration and heart rate rate enhance. Sweating stage is the last stage, and here will be sweating and temperature goes down to normal. Okay, so next we have the history of malaria, you can just go from it. Next we have the prevention, how we can prevent. We all know that if there is a mosquito present, so we should close our doors, and we can use even nets, okay, for the prevention of entry of mosquitoes. So by closing doors, windows, etc., to check entry of mosquitoes, we can use insect repellents to the to prevention from the mosquitoes bite, and sleeping under the mosquito nets, and we can use some chemicals to prevent killing of mosquito, killing of adult mosquito by some spraying. Okay, we should just avoid to kill them because they are also living organism like us. So we can use some preventions, like we can use the mosquito nets, and doors are closed, windows closed, etc. Okay, so next we have the therapy. We can use the some drugs, like these are the anti-malaria drugs, that is the quinine, chloroquine, so these daraprim, etc. So most effective drug is the daraprim, which kills the parasitic stage present in both liver and RBCs. Okay, so this is the all about the Plasmodium vivax. If you have any query, you can simply comment in comment section. So that's all about. Thank you.